Thanks for tuning in tonight to hear a little bit about the role of minor viruses in BRD. So many viruses have been associated with respiratory disease in cattle. And as you look down this list, you see names that you know well, and then maybe some other names that you don't know as well, or maybe have never heard of. But all of these viruses have been associated with respiratory disease in one or more ways. So some of them have been isolated, either the whole virus or nucleic acid from the virus, from cattle with clinical signs of respiratory disease, possibly in individuals or sometimes in multiple animals in outbreaks. Some of these viruses have been associated with BRD based on seroconversion at the time of an outbreak of clinical BRD. So investigators didn't really find the virus, but they found the cattle mounting antibodies to the virus. And so that does give some proof that the cattle have been infected, although it doesn't really necessarily prove that it was the cause of respiratory disease. And then for some of these viruses, it's been identified that they can lead to clinical signs of disease and gross and or microscopic pathology following experimental challenge. And so in cases like that, they might have even been used to fulfill Koch's postulates. However, it's important to know that all of these viruses can also be isolated from cattle with no clinical signs of disease. And that's more common for some of these viruses than others. Here's an abstract from a paper published a long time ago, 1980, but it's one of the studies that led to what we sort of understand today about which viruses are important and which are maybe not so important. In this study, they looked at over 1,500 calves that, in which 22% had been treated for respiratory disease and 2.5% died. Viral infections were identified on 540 occasions, and they identified PI3, RSV, rhinovirus, BVD, adenovirus, rhinovirus, and enterovirus. So they found all these different viruses in the calves, but only RSV, PI3, and BVD were significantly associated with disease. And so, of course, these are three viruses that we've talked about ever since. Here's a much more recent paper, 2016, where investigators used some new technology, metagenomic sequencing, to look for viruses in feedlot cattle. And these were feedlot cattle in Mexico and the United States. And they found the nucleic acid for bovine rhinitis A virus, rhinitis B virus, coronavirus. And these were the most commonly identified viruses of the 21 viruses that they found altogether. However, Influenza D virus was the one that was significantly associated with BRD. So again, they found 21 different viruses. 3 that were most common were rhinitis A, rhinitis B, and coronavirus, but the one associated with BRD significantly was Influenza D virus. And here is a more recent paper even yet, 2019, where investigators looked for viruses in feedlot cattle in Western Canada, in nasal swabs and tracheal washes of 116 feedlot cattle, and they found that the presence of Influenza D, bovine rhinitis A virus, bovine rhinitis B virus, and coronavirus, and also respiratory syncytial virus were associated with BRD. And so these are the kinds of examples of studies that are done that give us an idea that certain viruses may be associated with BRD and others may not so often, even though we find them. Now, when we talk about BRD outbreaks, it's not unusual for us to look for some of what we consider the major players, IBR, BVD, maybe BRSV, and sometimes not to find them. And so it may be that some of these minor viruses we'll talk about tonight are playing a leading role in those cases. And you have to ask yourself whether some of these minor viruses are disproportionately associated with outbreaks where the cattle just don't get sick enough for people to run diagnostic tests. And so that leads to our diagnostic labs having less opportunity to find them, leading us to believe that they're not really important when they may actually play a role. So tonight I thought we would talk about a little bit about parainfluenza type 3, bovine influenza D virus, coronavirus, bovine rhinitis virus, and bovine rhinovirus. Now, parainfluenza type 3, we might debate whether or not this is a minor virus. I brought it up tonight mainly because many people these days don't think of it as being very important. But we have some evidence that it may be important in some cases and might even become more important in coming years. Just a few words about the virus. It's in the family Parainfluenza, which means that it's enveloped and has a single-stranded RNA genome. And so the reason I mention this is that envelope viruses, all things being equal, are usually easier to kill with disinfectants and don't tend to persist in the environment as long as viruses that are not enveloped. And so that can be kind of practically useful. Single-stranded RNA viruses tend to vary more, and so often genetic variation is a method by which these viruses cause problems by mutating and evading the host immune response. Now, you might be interested to know that PI3 is in the same family, or this family Parainfluenza, BRSV, is also in that family, so PI3 and BRSV are related. We do find BRSV these days more commonly associated with disease, and you can generally cause more severe disease with challenge, although that's not always the case either. PI3 virus, interestingly, causes disease in many species, especially sheep and goats. Now, as I said earlier, many people don't think PI3 is very important anymore. It was one of the first viruses associated with BRD in the 20th century, and investigators found it associated with Pasteurella and led them to describe these sort of co-infection outbreaks that we recognize even still today, basically primary PI3 virus followed by secondary Pasteurella or Mannheimia infection leading to BRD. PI3 occurs as 3 different strains, A, B, and C, and they circulate in cattle populations. There were a couple of papers in 2017 that showed that the virus is starting to vary genetically from the strains that we have in vaccines. And so it suggests that common use of vaccines that contain PI3 may be inducing some pressure on the virus to change. And so that could mean that we'll be seeing more of it in the future. Antibodies to PI3 are widespread. That could be because we give vaccines that very commonly contain PI3. Seroconversion has been associated with about 30% of BRD outbreaks in some studies. However, in other studies, seroconversion occurs, but it's not associated with disease. So it's pretty common to find cases where the cattle seroconvert, but nobody ever noticed that they were sick. And so it may be that when it is occurring in association with BRD outbreaks, this is because of other things that were also happening to those cattle. So overall, disease due to PI3 alone is rarely seen outside of experimental challenge studies. If calves are challenged with PI3, you can induce some signs of disease, and it can even include more severe signs such as increased bronchovesicular sounds and wheezes over the lungs. PI3 infects epithelial cells from the nasal passages all the way down to the alveoli in the lungs, and they can even, PI3 can even cause syncytia, similar to BRSV. And it does damage the epithelial cells, leading to dysfunction of the mucociliary apparatus, and it can also suppress alveolar macrophage function. So it does have ways to impair the host respiratory defense and increase risk for secondary bacterial pneumonia, as we've discussed. And so this is just a photomicrograph showing an airway in the lung of a calf. Now, there are multiple syncytial cells here kind of pointed out by the arrows, and some others as well in this airway. And so this is just a reminder that PI3 can cause syncytia. However, this is actually a picture of a calf that had BRSV. So while they're similar, again, BRSV is usually more severe. Most multivalent vaccines contain PI3, and some of my friends in industry have told me that this is because if they took the PI3 out of the vaccines, they would have to have the vaccine approved all over again, and this is just too much trouble and expense, and so they just leave the PI3 in the vaccines. This widespread vaccination may be why we don't see much disease. We may have induced widespread herd immunity, and in fact, if you think back to those papers I described earlier in this talk where investigators looked for viruses in feedlot cattle, in the recent studies, they're not finding IBR virus very much. And so maybe the widespread use of IBR PI3 vaccines, which have been together in vaccines since the 1970s at least, is the reason we don't really see too much of either of these viruses causing respiratory disease anymore. I think we have induced some pretty good herd immunity in cattle with the vaccination, but that's just the theory. It's a little bit hard to prove something like that. It does lead you to wonder if we would see more PI3 if we stopped vaccinating for it. All right, now a little bit about influenza viruses. Of course, influenza viruses in most years are a cause of a lot of disease and worry in many different species. They're in the family Orthomyxoviridae, which means that they're enveloped negative-stranded RNA viruses, but the important thing is that they have a segmented genome. So flu is almost like it's got tiny little chromosomes because the genome is in something like 8 to 10 segments. And that means that they can reassort. And this means that exciting new types of flu can occur due to mix and matching of segments from different strains co-infecting the same host. So if you have an airway epithelial cell, like down here in the sort of bottom of this slide, and it's co-infected with two different influenza viruses at the same time, which does happen sometimes, namely, for example, influenza strain 1 that has its segments and influenza strain 2 that has its segments. If these two viruses infect the same cell at the same time, then what that cell can produce is a brand new influenza strain with a whole new set of segments that has maybe never been seen before. And this is what leads to the pandemics that we occasionally see due to influenza in people, when especially influenzas of birds and or pigs and or people mix in the same cell, and that leads to new viruses that people don't have good immunity to, or the pigs or the birds don't have good immunity to, leading to lots of sickness. Influenza viruses occur in 4 different genera, alpha, beta, gamma, delta, and these are then referred to as influenza A, B, C, or D. There are important genetic differences among these genera, so viruses in one genus can't reassort with viruses in another genus. Influenza A is the common one that affects humans, swine, poultry, also horses and dogs. Influenza B affects humans and occasionally other species. Influenza C affects humans, especially children, occasionally other species. Cattle have been shown to carry antibodies to influenza A, B, and C, indicating that cattle can be infected with these viruses, even though we don't think of influenza as an important disease of cattle, and we don't really worry about cattle being a source of pandemic influenza in people. They certainly can be infected with the influenza. And seroconversion has been associated with outbreaks of BRD when no other sort of major player virus has been found. The case I've referenced here is a situation where cattle were apparently infected with a human strain of influenza A. And so it does suggest that influenza A may rarely cause BRD outbreaks. Probably even more important is that if cattle can be infected with influenza A, it might be possible for them to be a sort of an animal species where reassortment with other influenzas could occur, maybe leading to novel new influenzas. And it may be that one of these will eventually become more severe in cattle, leading it to be an important cause of disease that we recognize, like is the case with dogs. 30 years ago, we didn't think of influenza as a cause of respiratory disease in dogs, and now it is. And so that could happen with cattle as well. Influenza C has been isolated from cattle with BRD, although the significance is uncertain at this time. But if you have a BRD outbreak, it could be that an influenza virus is the cause of it. Now, we've talked about influenza A, B, and C, but we need to spend a little bit more time on influenza D, because this is the influenza that is very commonly found in cattle and has been associated with BRD in multiple different ways. So it was first isolated from a pig in 2011. And so investigators then went to look for antibodies in pigs and also in people to see if this was going to be a new virus we were going to have to worry about. But the prevalence was pretty low in banks of serum that were tested. However, when investigators tested serum from groups of cattle, they found a very high prevalence of antibody, and then ultimately went on to show that cattle around the world are very commonly seropositive to influenza D. Most herds have at least one seropositive animal, and in general, in groups of cattle, 30 to 50% of cattle can be found to be seropositive. And although the virus was first isolated and recognized in 2011, a study evaluating sera from cattle collected in Nebraska in 2004 showed a herd-level seroprevalence rate of greater than 90%. So this goes to show that influenza D was quite widespread, probably at least 10 years before it was first identified, and even possibly earlier. It's interesting to note that a recent small survey found that in people with frequent contact with cattle, a small group of people that were tested, 91% were seropositive to influenza D, as compared to a small group of people that didn't have cattle contact, who showed only an 18% seropositivity rate. So this suggests that those of us who work with cattle may not infrequently be infected with influenza D virus. So far, it hasn't been identified, though, as an important cause of disease in people. There are two genetic lineages known as clades circulating in North America, and antibodies are partially cross-reactive. So the importance of that is that cattle could get infected with influenza D from one clade and then maybe within weeks be exposed to an influenza D of the other clade and still get fairly sick because the antibodies are only partially cross-reactive. Influenza D has been associated with BRD in many different ways. Seroconversion to influenza D has been associated with BRD in stocker cattle, and virus has been isolated from tissues of cattle with BRD, and some of this work was done here at Mississippi State in Henry Wan's lab when he was still here. Influenza D gene sequences have been identified in metagenomic sequencing projects and showing the virus to be associated with respiratory disease both in dairy calves and also in feedlot cattle. So various different investigators have shown a relationship between influenza D being present and cattle having BRD. However, experimental challenge of seronegative calves with influenza D leads to only mild disease. The virus seems to replicate and induce inflammation only in nasal passages and the trachea. And so, of course, this begs the question of whether the virus is important by pretty much like we discussed earlier with parainfluenza virus, causing abnormality of the epithelial cells of the upper respiratory tract and then predisposing cattle to secondary bacterial infection. And this may be more a problem when the cattle are also undergoing other sort of difficulties that decrease their resistance to infection. Some diagnostic labs do offer PCR for influenza D virus identification on nasal or nasopharyngeal swabs or lung tissue. So if you have an outbreak and you are wondering if influenza D is involved, you can identify it. So to summarize, influenza D has repeatedly been associated with BRD around the world. It leads to mild disease following experimental challenge, however, which suggests that the virus contributes to BRD only in combination with other factors. And there is no commercially available vaccine for influenza D in the United States at this time. Now, a few words about coronavirus, and I feel like this group, we've probably talked about coronavirus a good bit, so I maybe could have not talked about this tonight. In fact, we've probably all heard as much about coronavirus as we cared to in the last year, so I'll just kind of go through this part quickly. Coronavirus is also an enveloped single-stranded RNA virus, similar to the other viruses we've talked about tonight. So the fact that it has an envelope, again, means that it should be relatively easy to kill with disinfectants and not likely to persist in the environment. So I guess that's one way the COVID-19 pandemic could have been worse. If it had been caused by a virus that wasn't enveloped, then we might not be able to kill it on our hands and our work surfaces with alcohol as easily as we can. So I guess that's something to be grateful for. Of course, coronavirus is an important cause of calf diarrhea and winter dysentery in older cows. You may not know, but when a calf has diarrhea due to coronavirus, it will shed coronavirus from its nose too. So that complicates interpretation of nasal swabs in dairy calves. So if you're dealing with an outbreak of respiratory disease in dairy calves at an age when they're also likely to have calf diarrhea, usually kind of in the first sort of 2 to 6 weeks of life. It's really hard to say whether the virus coming out of their nose is a primary respiratory pathogen, because it might just be there because they are infected with the enteric version of the virus. So that's really important to keep in mind when you're working up dairy calf BRD outbreaks. We did describe high morbidity, low mortality BRD outbreak in pre-weaning beef calves in Georgia when I worked there. It was pretty remarkable. There were 180 calves that we sampled, and we tested swabs from 60 of the 180 calves. And what I thought was really noteworthy is that the fevers were really high in these calves. I think there were 10 that had temperatures of over 107, and the average temperature in 180 calves was 104.5, I think. So many of them were febrile, but only 1 out of the 180 died. And we tested 60 of the 180 nasal swabs, and we got coronavirus off 90% of the swabs. And we did not find BRSV or IBR or BVD. So that was an outbreak that made me think that coronavirus was an important contributor to BRD. Although, as I've said, we might have missed other viruses that we didn't think to look for. Lots of studies have shown a relationship between coronavirus and respiratory disease in stocker and feedlot cattle. Nasal coronavirus shedding has occurred in conjunction with BRD outbreaks in stocker and feedlot cattle when there was no diarrhea, so this is different than the situation with dairy calves. You can certainly have BRD with no GI signs in stocker and feedlot cattle, where you isolate a lot of coronavirus. One high morbidity, high mortality outbreak at least was described, where coronavirus and manhymia were both found together. Seroconversion was shown to be a risk factor actually as early as 2006. And antibodies being present at feedlot arrival decreased the likelihood of BRD in a study as early as 2002. So lots of evidence, sort of direct and indirect, that coronavirus is involved with BRD in stocker and feedlot cattle. Recent isolates are genetically divergent from older isolates, and there are some genetic differences between the isolates from respiratory cases versus those from GI cases. However, cross-infection between systems does seem to occur. So if you have the question in your mind, are the viruses that cause GI disease the same as those that cause respiratory disease? I think the best answer is to some degree, but not always. One thing about coronavirus that's been a little bit frustrating when it comes to talking about its role as a respiratory pathogen is that if you expose young seronegative calves to enteric isolates of coronavirus, they will very reliably develop diarrhea. However, if you challenge seronegative calves with a respiratory strain by a respiratory route, they do not get very severe respiratory disease. And lots of different people with a lot of experience in BRD microbiology have tried. And so it suggests that, perhaps like we discussed earlier with Influenza D virus, and to some degree Parainfluenza type 3 virus, that other factors also need to be present for coronavirus to cause important disease. Maybe you've seen this study or heard about it, a nice little clinical trial where stocker cattle were given intranasal calf diarrhea vaccine containing live coronavirus and also rotavirus. And there were also controls not vaccinated, so it was a controlled clinical trial. And the cattle that got the intranasal coronavirus vaccine did have significantly decreased BRD morbidity relative controls. So this study supports the concept that intranasal modified live coronavirus might help decrease BRD in stocker cattle and also in calves. And anecdotally, we've worked with some different cow-calf operations where pre-weaning intranasal coronavirus vaccine has been used to apparently help with pre-weaning calf pneumonia. Just a word about biosecurity. A recent study showed that infectious coronavirus could be found on boots and clothing 24 hours after personnel handled infected calves. So although I said earlier that the virus is not going to stick around for too long, it can persist for at least 24 hours on boots and clothes. So you want to keep this in mind, because that suggests that if you're moving from farm to farm or calf group to calf group over the course of hours, you certainly could transmit coronavirus on your clothes and your boots. So you definitely want to think about that and use proper technique to clean your boots and disinfect them. So there is ample epidemiologic evidence linking bovine coronavirus to bovine respiratory disease. However, the repeated failure of experienced researchers to induce respiratory disease with experimental challenge causes some persistent doubts about the importance of coronavirus. And maybe the fact is that coronavirus just can contribute to BRD, but it can't go it alone. That's kind of what I think is the situation. So it is one of these sort of potentially contributing factors that just can't cause disease by itself, but may tip cattle over the edge when there are other problems at the same time. Now, a few words about a couple viruses with similar names, and so they might be easy to confuse or overlap in your mind. Bovine rhinovirus and bovine rhinitis virus. Now you're thinking, who names these things? Well, they came out of the nose and they're a virus, and it was a cow, and so rhinovirus, rhinitis virus. That's probably what happened. They are similar. They're both in the family Parainfluenza, which means they're very small, non-enveloped, single-stranded RNA viruses. So I guess that's a common theme. Everything tonight's been an RNA virus, but these are the first ones that don't have an envelope. So these are going to be more persistent in the environment, not as easy to kill. The rhinoviruses are in the genus rhinovirus, so family Parainfluenza, genus rhinovirus. And so these are similar to the rhinoviruses that cause the common cold in people. So you could think of rhinovirus as the cow's common cold virus. In contrast, the rhinitis virus is in the genus adenovirus, and this is the genus that also includes foot and mouth disease virus. So that's kind of scary, but they're obviously very different in terms of the disease they cause. And we, as we discussed earlier, have rhinitis virus, or as we're about to discuss more, we do have rhinitis virus all around the place in North America, but we are trying hard not to have foot and mouth disease virus in North America. These viruses are not easy to isolate in cell culture. So in the old days when diagnostic labs were using virus isolation a lot to identify viruses, these viruses might have been missed. And when these viruses are given in experimental challenge studies, again, kind of a recurring theme tonight, they cause little to mild disease. Bovine rhinovirus, so the common cold type virus, was described quite a bit in the past. So here's a paper from 1969 describing the results of experimental exposure of calves to bovine rhinovirus, a paper from 71. Rhinovirus is isolated from calves with acute respiratory disease, and another from 74, isolation of rhinovirus from cattle in outbreaks of acute respiratory disease. So again, as we've been discussing, you can find studies where people identified these viruses, the rhinovirus associated with respiratory disease. However, there has not been much published since, and in studies where it is identified with, for example, metagenomic sequencing, it hasn't been associated with respiratory disease, unlike the case with these papers from the 1970s. And so it might be that these have just gotten to be so widespread that the cattle have developed herd immunity. In contrast, bovine rhinitis virus, so this is the one that's in the same genus as foot and mouth disease, but it has been found in recent years in cattle. So here's a paper describing bovine rhinitis viruses in U.S. cattle with respiratory disease, published in 2015 by Dr. Ben House and colleagues. And I believe Dr. House is now at South Dakota State and is continuing to work on bovine rhinitis virus. Bovine rhinitis virus comes in 3 different strains, rhinitis A, type 1 and rhinitis A type 2, and then also bovine rhinitis B virus. Genetic material from all 3 of these have been isolated in the United States. Recent isolates are fairly similar to 30-year-old strains, so it looks like cattle are not developing herd immunity to the rhinitis viruses. And bank sera from 15 herds in 6 states all contained antibodies, so the viruses are widespread. So here again is the same abstract I showed you earlier on, where these investigators looked at different viruses isolated, they looked for viruses in feedlot cattle with nasal swabs. They found 21 different viruses to be detected, with bovine rhinitis A and B being among the most common. However, these were not associated with disease. In this study, however, 2019 paper also used metagenomic sequencing of feedlot cattle. Here they used nasal swabs and also tracheal washes. They did find bovine rhinitis A and also bovine rhinitis B to be associated with disease. So yet another virus that can cause disease or can be associated with disease in some cases may be found in cattle that no one has noticed to have respiratory disease in other cases. So to summarize, many viruses have been associated with BRD in cattle. Many of these cause mild or no disease following experimental challenge. So what does this mean? Well, it may mean that they're not really important, or it may mean that they're important, but they can't be important alone. And I like to talk about these now and again because we do sometimes have outbreaks when we can't find the common viruses. And so I think just keep in mind that there are lots of other viruses that may have been involved that your lab wasn't set up to look for because they just don't cause severe enough disease enough to sort of provoke the development of diagnostic tests. But if herd immunity is decreasing the circulation of major players like IBR, for example, we may see more from some of these minor viruses in the future. So keep in mind that they're out there. All right, and so then I'll be here to take any questions if you have them. Thank you. Hello, thanks for tuning.
Thanks, Dr. Woolums. Do we have any questions for Dr. Woolums on this? I do have some questions because a lot of times when we investigate BRD outbreaks, a lot of times coronavirus is the only virus that we ever find. And you know, most people don't really think that coronavirus is that big of a player in respiratory disease, but I kind of beg to differ the point on that sometimes. And I was wondering kind of what your thoughts on that were.
Yeah, no, I mean, I agree with you. Again, my sort of angle on this is I try to read the literature and try to evaluate it as thoroughly as I can, sometimes get involved in research myself. And I think there are a lot of research papers that suggest that coronavirus is involved with BRD. But the problem is, and you know this better than I do, people have tried to induce it experimentally so you could get a vaccine labeled to control BRD, and people haven't been able to do that, right? And so, so what does that mean? Again, I think that when we give an agent into the airways, it's a very artificial situation. The cattle are kept in isolation, not allowed to get sick from anything else, and so you don't see disease. But you find common association in these cattle, in these outbreaks, and lots of other people have too. You know, not just you folks at Merck, lots of people have shown relationship between coronavirus, but not always, right? So some of those abstracts I showed you, coronavirus was isolated, but it wasn't associated with disease. And what does that mean? Some of you might be wondering, well, usually in those epidemiologic studies, they sample a bunch of cattle with BRD and a bunch of cattle without BRD at the same time. Not, you know, if you do it properly, it's essentially a case control study. And if you see a virus in the cases more than the controls, then that would be, oh, the virus is associated with BRD. If it is just as often found in the controls as in the cases, then that suggests, well, it's there, but it's not associated with BRD. Coronavirus in some studies is associated with BRD and in some studies isn't. And I've had the same experience as you, Harold, where we investigate an outbreak and it's the only virus we find. But did we look for rhinitis virus and rhinovirus and adenovirus and enteroviruses? No, we did not. So we can't say for sure that it wasn't one of these other viruses as well. So, I mean, I think, you know, we talk in epidemiology about factors that are necessary but not sufficient. I think in some BRD outbreaks, coronavirus is necessary, but it's not sufficient. In other BRD outbreaks, it's not even necessary.
So we do have one other question asking about vaccinating stockers for coronavirus. So I'll get your thoughts on that.
Well, as I always put that little study in there, I say little study, it was that paper that Paul Plummer and his colleagues published in 2004, so it's getting to be an older study, and it wasn't a huge trial, but it was a nicely designed trial, and they showed that when they gave intranasal calf diarrhea vaccine to stockers, they were less likely to get BRD. Now, it would be nice to see something like that repeated, right? I mean, if I knew of bigger trials, you know, kind of like some of the others that have been done for other vaccines that investigated, that looked at coronavirus, I would tell you about them, but I haven't been able to find them. So And as I said in the presentation, some of us here, Dave Smith and I and others, Dale Grotelution, many of you know, I got invited by Dale and Dave to evaluate some of these cow-calf operations in western Nebraska that just treated pre-weaning pneumonia year after year after year. And I mean, I think one of the producers got up and talked at the BRD symposium, if some of you might have been at in 2019, and he basically said coronavirus vaccination changed our life, right? I mean, it was even more dramatic than I would have said. But because in those cow-calf operations in western Nebraska, we had recurrent pre-weaning BRD. We did have an opportunity to swab some calves when they were like a month old and no coronavirus found. And then they had the outbreak when the calves were sort of 2 to 4 months old, and we found coronavirus. And so again, you know, it's circumstantial in a way. We don't know for sure, but that was what led us to say, why don't you try intranasal coronavirus vaccination? And you know, this is extra label. I mean, I probably shouldn't even be saying this in an industry-supported talk because I know we're not supposed to be pushing extra label stuff. But those two ranches where they had treated a lot of pre-weaning pneumonia for many years felt like once they started giving coronavirus vaccine intranasally, and let me emphasize that I do think it's important that when you're dealing with pre-weaning pneumonia, I think the calves need to be vaccinated twice. And that's before weaning. And that's a big pain, right? Who in the world would want to vaccinate pre-weaning calves twice? I think it's only people who have treated a lot of calves with BRD pre-weaning, right? And so you have to be motivated to do that. And I don't even think everybody should do it, right? If you don't have pre-weaning BRD, don't worry about vaccinating the calves pre-weaning. But in these ranches where they had been very frustrated by years of treating pre-weaning BRD, getting the coronavirus intranasal into them when they were a month or two old, and then getting a second dose into them. Usually what we try to get them to do is get another dose into them right about the time they were expecting the pre-weaning pneumonia or summer pneumonia outbreaks. Again, I wish I could do a big clinical trial to provide some really high quality data to support what I just told you. But it did make me happy to have two producers tell us, look, we feel like we've dealt with our problem. You know, one thing I will say, to be honest with you, maybe it wasn't just the coronavirus. Maybe it's getting the calves all up together twice before they were having their summer pneumonia outbreaks, so their own endogenous other viruses that we aren't even measuring can get, you know, mixed around among them. I mean, I think that's one thing Dave brought up, and I think it's a really good point. So,
any other questions for Dr. Woolums? Comments? Well, Dr. Woolums, we appreciate you participating and value your insight.
Thank you, guys. Thanks very much for inviting me. I love being involved with this meeting, and thank you, Harold, and Ronnie and everybody else at Merck for supporting it again.
Thank you.